Thanks very much for coming to today's seminar with Matt McCary, who's coming to us from Rice University. I've heard from a lot of folks about how exciting it's been to talk with Matt and have him here, and I fully agree. I met Matt last summer when he got the award for Early Career Fellow from the Ecological Society of America. And so I've followed his work very closely for a number of years, met him for 30 minutes and thought this is someone that we really need to come visit us at the University of Michigan. And it's true, it's been great. Matt got his undergrad degree at North Central College in Naperville, Illinois, home of Candace Parker, basketball fans. He got a PhD with David Wise at the University of Illinois, Chicago, and then did a postdoc at Wisconsin and a postdoc at Yale with Bob Schmidt, who got his PhD here, actually. Since 2021, Matt's been an assistant professor at Rice, leading a really cool series of projects that we'll hear about today that are really about the intersection of global change, ecology, community ecology, and ecosystem ecology. He's also done a ton of really important DEI work in a variety of different avenues that I hope you get to hear from or hear about today. So it is a real treat to have you here, Matt, and looking forward to your talk.
Thanks, Nate. Can everyone hear me? My voice doesn't carry that well, so I was like, a microphone is a blessing, so I'm very happy about that. But yeah, thank you, Nate, for the invitation. It's been a great time today getting a chance to interact with the department, the grad students, the faculty. It's been a really set of rich sort of conversations I had today, and the work that y'all doing here is inspiring, and very thankful for the time that I had today. But yeah, so today I'll be talking about sort of some of the sort of projects that I get very excited about that I've done previously, which is kind of getting at this idea of how global change can really impact the soil biodiversity and associated ecosystem functioning. So I kind of have two case studies that I've done, and I think that the work that I'll be going forward would actually kind of be more or less in this vein here. And the title of my talk is Disentangling Soil Food Web Dynamics in the Era of Global Change. See if this is, all right. So this is kind of an American hardwood forest here that we often think about, especially at this, especially in Michigan. You have, you know, if you look at this, oops, deal with this here. Yep. Yeah. All right. Yeah, so we have our American hardwood forest. When you kind of walking through this forest, typically being bipedal, you know, thinking about what humans are, you get really drawn by the sort of the vegetation, right? And think about the greenery, think about the different overstory plants, the understory plants, and it's very beautiful, right? And we often kind of get enamored by that. But if you actually took a moment to think about what you actually see, you quickly begin to realize that a lot of that is actually filled through the below ground food web, which I will be talking more about. And in fact, everything that you see above ground, about 80 to 90% of that was at some point recycled through the soil at some form and fashion. And so basically you have a fundamental idea that everything you see above ground is actually related to what goes on below ground. And so part of what I really try to do in my research program is to really disentangle those interactions so that you can have a greater appreciation for what you see above ground. All right, maybe that, okay. Is it stuck? Oh, okay. Well, if it's too much of a problem, okay. So if you take a step, take a moment while you're walking through this American hardwood forest, especially in the sort of the fall when the leaves start to drop, and just kind of look beneath your feet, you will see an ecosystem that looks like this here. You have a lot of litter that's dying, and you know, oftentimes you kind of underappreciate what this ecosystem is, and there's a lot of players that sort of occur in this particular ecosystem that are super important for what ultimately gets cycled and ultimately the ones that end up, the plants that you see above ground. So just to kind of talk about some of the players here, we have All right, bacteria. So we got bacteria. So bacteria, they're going to be very important for breaking down organic matter into plant available nutrients so then plants can actually uptake. We have various sorts of fungi, so saprophytic. We have mycorrhiza fungi. They are super important for also releasing or allowing for plants to uptake nutrients and so forth. And they actually form very important mutualisms with plant communities. We have a centipede here, which is a major predator in the soil. We have millipedes, which are macrodetritivores, so they actually break down the large organic matter, such as leaflet, into smaller bits that then allow for bacteria and fungi to finish the decomposition process. We have a predator here, or scavenger, which are harvestmen, or daddy longlegs. We have lineatheid spiders, or ground spiders, which are another major predator group. We have slugs. We have my personal favorite, columbella, which are very cute, and they are very important in terms of fungal grazing and decomposition. We have mites, which are incredibly abundant and diverse in their own. They have predators, fungivores, bacteriivores, have roly-polies or wood lice, another macrodetritivore, and we have earthworms, which in some ways are ecosystem engineered. And so you can just take a moment and really appreciate the different players and the diversity of this system here. And one can quickly realize that each one of them kind of serve a function, particularly regarding how fast or slow nutrients are recycled. And the interactions that occur between these different groups can really impact those rates of nutrient cycling. So basically, there is a very simplistic form in which we kind of organize this sort of soil community here. And I'll kind of walk you through a very basic, overly simplified, but a basic form of how we think about this system. So typically we have your hardwood forest. We have the leaf litter that is dropped and it kind of creates this pool of organic matter. That's going to stimulate the microflora, in this case, bacteria and fungi. And through their interactions, they can actually release nutrients back into the system that allow for plants to uptake and kind of really generate sort of dynamics that plants can then thrive. We then, what I call primary consumers, which feed on the microflora, both bacteria and fungi. And through their interactions, they also can impact nutrient cycling. Then last but not least, we have predators that can impact the abundance of primary consumers, and they can also mediate nutrient cycling. What we typically think about this system as being bottom-up control, meaning that the amount of organic matter going into the system is really what fuels how big or small these particular pools are, and then how that would relate to nutrient cycling. So in one case, you can imagine if you have a case where there are fewer resources going into the soil food web, each one of these pools are going to get smaller, and these theoretically would see slower rates of nutrient cycling. And then on the contrary, if you have more nutrients being filled, each one of these pools would get bigger, and theoretically there should be more nutrients being released, and in some cases faster. And so we think about this as being a sort of a bottom-up food web, although there are certainly some nuances to that as well that I try to disentangle in my research program. Even though we have some ideas about how this system functions, and that's kind of our theoretical framework of how we think about the system, we also know we don't know everything about it, and that's an overly simplified version of this particular food web. We also know that there's a lot going on globally that can threaten how this particular system functions and how it relates to ecosystem functioning. And so we can think about sort of habitat destruction, or thinking about this in terms of actually changing the amount of resources that goes into the soil food web. Think about invasive species, the quantity of resources going in can drastically change as a function of invasive species or invasive plants in this case. And then we have sort of deposition or pollution that can also change the type of resources that enter the food web, and subsequently the type of nutrients that will be recycled, and so forth. So basically, what I'm really interested in my research program then is to be able to, for one, preserve and sustain these communities, these ecosystems. We really need to first understand how they sort of respond to resource changes as a consequence of sort of global change. So the outline of my talk, I'm going to present two case studies. So the first case study would be looking at a sort of case where you have an invasive plant that comes in and actually limit the amount of resources that are in the soil, and then think about the cascading effects up the food web. And then I present a case where you have this subsidy that actually substantially increase the amount of nutrients going into the soil food web, and then think about what are the cascading effects of the food web as well. So in this first case, thinking about famine, thinking about fewer resources going into the soil food web, what are the implications for that? And before going into that, I want to just take a step back and think about invasive plants in general. So typically, invasive plants reduce the diversity of invaded ecosystems. So this is in the case of Japanese knotweed. I can give you a clue that knotweed is not supposed to look like that. It's consuming this young lady here. And invasive plants in general tend to have this sort of response to new environments where they become really problematic and can really degrade the diversity of these ecosystems. We have invasive Phragmites that create these monocultures in wetland communities that can really erode the diversity of these communities. We have common buckthorn, which also occurs up here, to create these very dense thickets that chokes out all the native plants and can really erode the native diversity. And then we have kudzu, which is a sort of vine that we get down in the south that can just blanket the whole ecosystems. And so, we think about invasive plants in particular, we generally think about them as sort of changing the biodiversity of these systems, both in terms of plant competition, but also one can imagine the food web as well will be impacted. All right. So one can also think about the resources that would be changed as a function of these invasive plants coming into the system. So in this case, it can certainly influence the detrital input. So when leaves fall to the ground, the content and quality of that litter could be super problematic and different from the native community. This is kind of blurred here, but this is common buckthorn. What's really interesting about buckthorn is that it actually has very nutritious leaf litter. And so when it actually falls, it actually decomposes faster than like oak litter. And so what happens is that it creates these like bare ground situations. And you can imagine that's going to have a pretty negative impact on the soil community as a function of this invasive plant here. You could think about changing disturbance regimes. If you think about fire or lack thereof, can really impact the sort of resource communities for the soil. It can also alter the microhabitat. If you got a lot of shading or these thickets that can really change the microhabitat, that can really be important for the soil community. And then in some cases, you can have changed soil chemistry when you have certain invasives like black locust that comes in as well. So this is kind of what I did some years ago now, but one of the things I was really interested in is like, okay, we know that invasive plants can be problematic for the diversity of some of these ecosystems, but one of the things I was really interested in is like, what does this mean for the food web associated with these ecosystems as well? And so I did a global meta-analysis to kind of look at how sort of the primary and secondary consumers are impacted by invasive plants. And I did it by looking at the green food web, which is anything that's above ground or plant-associated webs. What we found in general is that at least in wetlands and in forests, they tend to have a negative impact on the primary consumer, but no effects in sort of grassland ecosystems. But what was really interesting to me is that looking at least in the woodland, which is this middle panel, they tend to have an increase in impact on secondary consumers or predators and so forth. And so that kind of spurred some ideas of like, well, how can you have these contrasting effects? Is it because you see a negative impact on primary consumers because these invasive plants are creating habitat for predators, and that's going to have a negative impact? Or is it something different about how invasive plants impact the food web? And so this bore out some ideas or some case studies that I wanted to kind of follow through to see what those impacts are. And particularly in the brown food web, that was what I was really particularly interested in. So this brings me to garlic mustard, a plant that I think I appreciate dearly. So garlic mustard is this really interesting plant. It's a biennial plant, and first year it creates these very inconspicuous floral rosettes that in some ways can be a little bit hard to identify. In the second year, they kind of bolt and have these second year plants where they kind of go into fluorescence, and they then senescence in maybe about July and August in some cases. And then, each plant is pretty prolific. So you can think about each plant can produce up to, I think, 3,500 seeds, and they can remain viable for a long time. But what's really interesting about garlic mustard is that they actually have antifungal characteristics and glucosinolates. And so in the second year, the thought is that once they go into senescence, the cell walls break of garlic mustard, and they kind of release these glucosinolates into the soil. And these glucosinolates are antimicrobial. And so there's been a lot of evidence to actually show that in garlic mustard patches, you get less fungi or mycorrhizal fungi specifically when there's a high abundance of garlic mustard. And as I alluded to, fungi is a major food resource in the soil. And so it got me thinking, well, what happens when you have these crazy invasions of garlic mustard? Do you actually see changes in the soil food web? So this is kind of around the Chicagoland area, and this is kind of what an urban forest would look like, at least a relatively healthy one. And you can see that there is a lot of sort of leaf litter there, some diversity in the understory and so forth. And that's what an urban forest in Chicago would look like. This is what an urban forest that's invaded by garlic mustard in some way, in some areas, can look like in terms of garlic mustard invasion. And one can imagine that if each one of these plants are secreting glucosinolates and is going to change the sort of composition and abundance of fungi, one can imagine that's going to have a really pronounced impact on the soil community. So to kind of think about the theoretical framework for this particular study, we have the native plants, they have a positive association with the organic matter in a native sense. That's going to stimulate a positive influence on the microflora, in this case fungi. That's going to have a positive impact on fungivores and their associated predators. Income garlic mustard, we definitely know for many years now that there's a competitive advantage of garlic mustard over native plants, and so we'd expect there's going to be a negative relationship there. We also know that, through the literature that, there's going to be negative impacts of garlic mustard on the microflora. And then one can then suspect that there's going to be negative indirect impacts on fungivores and their predators. And that was kind of what I was interested to see what happened and kind of design some experiments to kind of tease that apart. So to do this, I selected sort of 5 sites that had naturally occurring populations across the Chicagoland area, and these areas span in very, very heavy densities of garlic mustard. And they also span long-term populations of garlic mustard, so they were invaded at least 10 years when I started this experiment. And then I kind of set up in the studies, sort of observational study across these sites where I had garlic mustard patches, and then within these garlic mustard patches, I had selected 5 areas where the garlic mustard was in some heavy and sort of density. And then on the periphery, I had areas where garlic mustard did not exist at the time. And it's very hard to document if they ever had garlic mustard invasion, but at least for the time of the experiment, they did not have any garlic mustard whatsoever. And so across these 5 sites, I had 10 plots per site, 50 plots total, and this was done over 2 years. And within each one of these plots, they were 1 by 1 meter in scale. I took soil cores, and so I looked at mycorrhizal hyphae. So I actually measured the amount of hyphae in each core. Also did some genomic sampling as well, looking at the fungal composition. I extracted soil arthropods, looked at columbella, mites, and so forth, and also measured the plant community composition across these plots, just to kind of sequence, just to sort of monitor what type of communities were across these various plots. And this was done over the course of 2 years there. So at a very broad level, I was just fundamentally curious to know across each one of these trophic levels, was there a signal in terms of the difference in fungi, predators, and they prey? And interestingly, when we look at the hyphal length, we saw that there was certainly much higher hyphal abundance in the control plots that did not have garlic mustard compared to the invaded plots, and those are sort of 95% confidence intervals there. And so it was kind of almost twice as much in the control plot as the invaded. When looking at the fungivore abundance, and these are mostly columbella and orbatic mites, we saw that there were more fungivores in the control plots compared to the invaded plots. We see a trend, although it's not a non-statistical difference there between the control and invaded regarding predators. And I will say that this is just a very coarse measurement. It really doesn't take into account the differences in how abundant garlic mustard was across these plots, but just thinking about the control versus invaded across all these different sites, we were just curious to see if there was a density impact. And you can see for at least the fungus and the fungivores, there were a signal, but not for predators there. But certainly was not the best way to really tackle this question. So I did a multitude of the different statistical techniques to kind of get at that. As I mentioned, we looked at the fungal composition. So this kind of reflects at the end of the experiment, we took soil cores, extracted genomic DNA from these plots and measured fungal composition. And what we found is interesting that, so this is an ordination plot. And so each one of these symbols reflect a community of fungi. And the dark circles are the invaded plots, and the control plots are the uninvaded plots. And this is one plot. This is actually showing the strongest relationship we saw. So across all 5 sites, they saw very similar patterns, although the strength of that pattern was different, and that's why we had a site by treatment interaction there. I'm just showing the one that showed the strongest pattern because it makes it a little bit more obvious to see what happened there. And one can see that the sort of control plots are kind of scattered on the left axis on this first axis here. And then we see that the invaded plots are quite different. What's also very interesting is that we see that orbatic mites, which was the most common fungivore in our experiment, were more associated with the control plot, and predators seem to kind of be all over the place, kind of depending on the site. But once again, it's kind of re-emphasized that there's at least a signal of the fungal communities and their fungivores as well. Across all 5 plots, we saw similar patterns, but this one was the one that showed the most important one. To kind of put everything together, we did a structural equation model to really account for the different densities of the garlic mustard, the different densities of the fungivores, predators, and the plants as well. And so to really see how all this kind of fits together here. So I'm going to walk through sort of some coefficients here that are very similar. So think about 0 being very weak in terms of the correlation, 1 being very high. And then I have bolded arrows to kind of indicate ones that were really obviously strong there. So getting back to it, so we had our native plants, have our organic matter, and then we had, this was not significant, but we did see a positive influence of native plants and the sort of mycorrhizal hyphae in the soil that we found. We did see that the amount of fungi had a very strong relationship with fungivores. And we also found that there was a very strong relationship of fungivores and predators. We saw that garlic mustard density had a negative impact on native plants. That's not surprising because that was part of the design in place. But we did see that there was a negative relationship with garlic mustard and fungi. And subsequently, we got indirect negative impacts of garlic mustard on fungivores. I would say that even though these coefficients look pretty low, we're pretty confident with like follow-up experiments that I won't show here, but we basically were able to show that it actually was the suppression of fungi that leads to those negative indirect impacts. But here, we were able to kind of more or less support the initial framing that we have for this particular study. So the take-home message, plant invasions can have really pronounced impacts on soil communities. And this, in this case, was an example that changes in the soil food web structure was a response to a fungal suppression, in this case by garlic mustard. And so, we kind of conclude that, these effects can really result in changes in native plant abundance and cover more broadly. So now switching gears, the garlic mustard was a case where we have fewer resources going into the soil community and soil in general. Here we have a very different sort of idea of how that may play out. So going back, think about sort of the theoretical framing for this particular study. We often think about ecosystems as being bounded, but in reality, especially at the periphery, they actually interact with other ecosystems. And in some cases, those resources that are exchanged can be really important for the structuring of the ecosystem that are receiving those resources. So in this case, you can imagine that this riparian forest, the leaf litter and leaching from the forest can actually release into the river. The river then had more nutrients that can stimulate algal growth and their associated consumers. And so you have an abundant aquatic ecosystem. And in this case, you can think about aquatic insects like mayflies or midges can then influence and integrate themselves on the terrestrial landscape. And so you have this cycle that really can impact both the terrestrial and aquatic systems. And so we kind of think about them as being bounded, but in reality, they are very reliant upon one another. A very classic example of this would be sort of the salmon migration that happens about October, November in the Pacific Northwest. So you got the sockeye salmon migration. They kind of are in the open ocean. They then migrate up to their spawning grounds in the river. And in the process, they occur and they actually become sort of a grisly feast. In some cases, you can think about the birds of prey that feed on them. And what happens is that, you have subdominant males that would kind of grab a sockeye salmon and then retreat in the riparian forest and just kind of leave a carcass that is there to deposit and fertilize the riparian forest in that case. And in some cases, there's some evidence to suggest that up to like 30% of the nitrogen that you get in these riparian forests in the Pacific Northwest have some signature that's related to a salmon carcass. And what ends up happening then is that when you have a riparian forest that is very productive, it actually helps the river and its productivity that then allows for the sockeye salmon to basically become more robust in their populations as well. So you kind of get this cycle where these two systems are relying upon one another. So taking the same idea, there's this very unique lake in northeast Iceland called Lake Mývatn. And so this was this very unique system that I had a chance to study as an NSL postdoc at the University of Wisconsin. And what's really interesting about Lake Mývatn is that at this given latitude, you get a lot of sunlight. It's a fairly large lake, and it's also very shallow. And so what ends up happening is that you have a very productive benthic or benthic algae population that then stimulates this very interesting species of midge that can create these enormous swarms in and around Mevat here. What's really interesting about Mevat is that at this location in northeast Iceland, what should be low-lying shrubs, very slow-growing vegetation, you get these very productive grassland ecosystems. And so as colleagues and I were thinking, well, is it possible that what's coming out of the lake is actually creating these grasslands that shouldn't theoretically be at this area in northeast Iceland? And so we've studied this Tanitarsis gracilensis. It's a midge species that occurred in Mevat. It basically has a very more or less typical life cycle of an aquatic insect. So it kind of goes through a series of instars. They then pupate as adults, and once they emerge from the lake as male and females, they then create these mating swarms on the adjacent terrestrial landscape, and actually can do some really interesting things to the terrestrial side of things as well. So I was particularly interested in understanding how disposal of resources from the aquatic side of things, how that could really impact the terrestrial landscape. And so just to kind of demonstrate the sheer abundance of what's coming out the lake here, this is definitely, this is all midges that you see here. These are specks of midges, it's not dust. So they create these very large swarms. So the males and the females create these mating swarms. And so the thinking is that the females return back to the lake to lay the eggs, and the males just stay behind and just die on the landscape. And there really isn't a top predator, and so they can actually accumulate a lot of biomass on these landscapes here. And so they usually have 2 generations. So they have one around like the end of May, early part of June. So we have the overwinter population that comes out, and they could be in really large numbers. Some estimates have suggested that in the first 50 meters of the lake, you get what would be equivalent to like a humpback whale in terms of biomass. So you can just think about that being slapped on the landscape. And each one of these carcasses are very rich in carbon and nitrogen. And so in an area that should be low nutrients, you get these midge carcasses that then can release these really vital nutrients that can really change the productivity of the system here. This is probably one of my very favorite pictures from Iceland here. This is a picture of Karen. She was one of the undergrad interns we had that year. Karen is like 6'1". She got these very long hands and fingers and things like that. And so this was actually after a midge emergence that we had. You can see that half of her index finger is submerged in midge carcasses. And you can imagine that all the nitrogen, all the phosphorus, all the macronutrients that occur in each one of these, you can imagine how that can really have an impact on a system that should be low in nutrients in general. So part of my research then was to really try to understand what was going on below ground to really stimulate these changes that we see where it should be a heathland vegetation. We got a system that's dominated by grasslands around Mevat. And so we have our heathland sort of community here. We have the organic matter. And so what happens is that, you know, there was some previous work before, like right before I started the postdoc. We saw that once you have a heathland, you add midges, you get a grassland community. We also have done some work here. I won't show all this work here, but we also was able to demonstrate that once you add midges, you get an addition or a higher abundance of primary consumers and their predators. But one of the things that we really didn't get a chance to tackle was understanding how the microflora respond, and subsequently, what does that mean for nutrient cycling? So effectively, how do you add midges to the system? How do you get a grassland system in a place where you should be seeing heathland? So to actually go about testing that, we had this sort of more or less control site. It was maybe about less than 5 kilometers from Mevat. That was a lake that actually had very low populations of midges. In fact, almost none came out of this, which is pretty interesting. So it kind of served as a control site. And what we had was what would be a classic heathland, and we did a midge addition experiment. So we kind of set out basically experiments where we added midges to half the plots and no midges to the other half. And this was a site that basically had no background midges whatsoever. And these were very, very small plots, 1 by 1 meter. I would say this site was incredibly hard to get to. It wasn't one that you can just drive up and kind of venture out. So that's why it wasn't as big as one would think it should be, but we definitely had a robust sample size. And based on calculations that come out of sort of MeVOT, it was about 100 grams of midges per meter squared that come out of MeVOT. And so we added about 100 grams of midges in these plots to kind of represent what would theoretically be happening at Lake MeVOT. And so within each one of these plots, we basically measured the whole miniature ecosystem. So we looked at plant community composition, we looked at litter decomposition. For litter decomposition, we had decomposition bags where we just had grassland litter, threw them in there at a known amount, and then harvest that at various points throughout the experiment to kind of get a rate of decomposition. We looked at soil microbes where we basically did phospholipid fatty acid analyses to look at the microbial composition and their abundance. And then we did soil CO2. So we took soil from each one of these plots and did incubation studies and measure how much CO2 came off of these plots, off the soil, to get a measure of microbial decomposition. And that's what we were able to do in this particular study as well. So it was really fascinating that just after 2 years of experimentation, you can see on the left plot, left panel there's just a control plot. So you see that there's typical heath vegetation there, and the grasses kind of occur in relatively small abundance here. But just after 2 years, we were able to recover a big sort of response in grasses here, where they become the dominant category of plants in this particular system. And it was a pretty obvious sort of response there. And so it's very fascinating, like even at this control site, just by adding midges, you can see that it becomes a grassland pretty quickly. When looking at sort of the amount of decomposition, and this is really fascinating because we use grass litter, so it wasn't like the decomposition of the midges, but we actually used grass litter in these plots. And what we found is that just by adding midges, you increase the decomposition by 2.5 times effectively. Certainly some variation across that, but we're very confident to say that they are quite different. And so it's really speeding up how fast decomposition occurs in the system when you add midges. So then this leads us to like, well, we know that you add midges, you get a grassland habitat, you get faster decomposition. And so we were wondering, was it the microbes? Was it the arthropods? Like what was really responsible for decomposition being sped up? Again, thinking about the nutrients being released that allow for these fast-growing graminoids and grasses to really respond. So there were a whole suite of measurements that I talked about, but what we saw is that the microbial decomposition was much higher in the midge addition plots than in the control plots. When we looked at the fatty acid analysis, the sort of microbe population, both bacteria and fungi, increased by like, I think 40%. And then we were able to actually see that the microbial composition was also very different in the control plots compared to the midge addition plots. And so we were able to kind of infer that it's most likely when you got midges, they stimulate the microbial community that then allows for the release of these nutrients, and the graminoids, because they are fast responding, fast growing, they respond and become the dominant plant community when you add midges to the system here. So to kind of put it all together, so what we saw is the microflora, their abundance increased 1.5 times when we add sort of midges. and then it increased the nutrients by over 2.5 times. And then that's how you kind of get a heathland to a grassland when you add midges in this particular system. And last but not least, this is kind of something I'm currently working on too. So we actually was trying to measure how much carbon and nitrogen cycling was happening when you actually add midges to the system. And so we working with John Moore at Colorado State University to kind of do some modeling on this here. And what we find is that there are huge changes actually. And so we saw that N cycling and C cycling over 10 orders of magnitude different when you add midges to the system. Yeah, oops. And what you can see is like the C flux is about 5 here, and then you get like, I mean, the numbers are incredible when you compare that there. And what's really fascinating is that these numbers actually, if you can see, are much more than what we actually added in midge carcasses. And so what this is showing is that when added midges, you actually stimulate a lot more decomposition and fluxes, more so than what's already there. And so it's actually some of the labile and recalcitrant sort of forms of carbon are also being sort of accessed and respired and actually decomposed when you add midges here. And I should say that you actually see an increase in trophic levels when you add midges, and so you get a more complex food web when you have midges, and the amount of sort of nutrients that are being cycled through here was just an enormous, you can't even really put it into words how different they were when you add midges. So this is based on the field research, and so this is kind of the modeling that came out of that. And so I'm hoping to be able to get that out sometime soon. So to kind of sort of put this all together, so the disturbances that affect resource change can really affect community structure and function of the soil. So in this case, insect carcasses can really increase decomposition rate, and then that can have sort of impact on the plant composition as well, and then microbes associated. We also show in the first example that removing fungi through allelopathy in terms of garlic mustard can actually change the soil food web structure. And in that case, we saw fungi and fungivores reduced. Even though the predator abundance didn't really have a signal, we do suggest that there's a change to the food web and associated interactions with nutrient cycling, presumably. And all this to say, all the, I think, really interesting work that I was able to do, we could just see soil food webs are just generally important for recycling resources and affecting the structure of these ecosystems. And we should be doing more to kind of really tease those interactions apart. So I'd like to acknowledge all the awesome mentors and interns that allow for me to kind of do the work that I presented today. And with that being said, I'll be happy to take questions.
So there's time for questions. I should have said this beforehand. There's not an on-campus happy hour after the talk. Instead, Julia and Olivia are taking a crew of folks to Red Hawk after this, so there'll be more time for people to talk. I also have to go get on an airplane, so I'm not running away because I'm offended by your talk. I thought it was really, really great. But if I do leave, thanks again for coming. It's great to have you.
Yes,
sir. Questions?
Thank you for a very interesting talk. So in the case of the fees, are you saying that there are more carbon released in the atmosphere because of the increased microbial activity?
I guess in general, yeah, that's true, yeah. I mean, the scale is probably not going to be a problem from climate change, but I think that certainly one can make the argument that more CO2 will be respired when you have these plots that are, I guess, getting a lot of midges effectively, yeah.
In the first case, the famine, would that have a reverse effect?
Yeah, no, that's kind of one of the biggest regret of that, is that I didn't really get a chance to link that to the ecosystem function in the way that I wanted to. But one can suspect that in that case, if you have less fungi, it probably will have a slower impact on decomposition, so probably less decomposition occurs where there are a lot high density of garlic mustard. And that way, probably less sort of carbon would be sort of respired. But that just would be me making an assumption. I don't have any data to kind of back that up.
Thank you.
Hi, great. I was wondering, maybe I missed it, like when you measured the decomposition rates, like what was the time point you used at the end of 2 years? Or was it sort of like temporary?
Yeah, no, thanks for clarifying.
Like alternative stable state or like
Yeah. So actually, the way that I did this here is that I had a number of litter bags that I put in each plot, and then at each sort of, I think I did it 4 times where I extracted the litter bags and measured sort of the amount of decomposition that occurred, and then I was able to kind of fit a decay rate across each plot, and that's how I kind of determined the decomposition rate. And there was a second part of that question? Okay. Yep.
No, go ahead. Maybe, I don't know if you've measured this or not yet, but I was wondering how long the effects of like the midge booms last on the grasslands. Like if there's a year where there's something wrong, like midges start having to all the plants die, or is that like just 3 years?
Yeah, no, I didn't actually do this work, but my postdoc advisor, Claudio Graton, actually ran an experiment where they did midge additions for like 5 years consecutively, and then they stopped adding midges, and then they actually was interested to see how long that effect lasts for. They basically ran out of time on that grant, so it stays a grassland once it becomes a grassland, and it actually, interestingly, these midges, what we think is like a slow release pellet, and so like they just get there and they just consistently releasing nutrients that then once it's in a given plot, they just recycle through that the whole community, and it actually persists quite a bit. Yeah.
So I was wondering if you took a step back from your plots and thought about the midge addition versus the aquatic system that's adjacent to it. So
Yeah.
you're moving these nutrients from the aquatic system into the terrestrial system.
Yeah.
Does it then go back into the aquatic system? I mean, is that like a tight coupling?
Yeah.
Or are you thinking that you're getting, because you're getting an increase in decomposition on the land, that you're starting to get a loss from your terrestrial system into your aquatic? Like how do you think that's working? Or do you have any ideas about that?
No, that is a phenomenal question that I think would probably be, if I were to continue this work, that would be like the NSF grant that I'd be writing up. It's like, what would be the impact of the midge? Because one would think that it's all of those midges, like even though they are really having an impact, a lot of that probably is leaching out right back into the lake. And that then keeps the population big as it is. And so you have this cycle that more or less they depend on one another. And so, but I can only speculate. I don't have any data, but that is something that I'd be very interested to talk more about. Yep.
Yeah, nice talk. This is kind of maybe a dumb question, but like, where does the grass come from?
That is a great question as well. We know that they kind of persist in low populations, so they kind of hang on, I guess, in this geographic region in Iceland. And then the moment that there is any nutrients that become readily available, they just like respond like crazy. But fundamentally, we don't know where they come from.
Native grass?
These are native from what I understand. They should be native to Iceland. One thing I should say, which was also very interesting, is that around Meebot, because they have the productive grasslands, they actually graze and farm on those grasslands. And so the local communities are actually dependent on these grasslands around Meebot. And the Icelanders call it midge grass. It translates to midge grass, but they call it mee grass. So they really know, they also know that these midges are really important for sustaining those grassland communities.
Do you guys see any long-term changes of like nutrient concentrations in these lakes at all, or is there a trend towards more grasslands or dwindling?
Yeah, so that's well, one thing that is smart on Iceland's part. So this is like they protect this area, and in fact, no one is allowed to even go in the lake without permits and things like that. So it's not like a heavy human pressure there, and so for right now, they are self-sustaining, and one can go there and expect to see these midge populations do what they do. But who knows in the next, especially with climate change, if that's going to persist or not.